he did his postdoc at UCL in London, studying MRI changes linked to cognitive deficits in multiple sclerosis. And 2015, he did a fellowship at Cardiff from Welsh Government using MRI to determine structural and cognitive problems linked to stress. Currently, he's the senior lecturer and deputy head of Division of Psychology and Human Resources at Uni of Manchester. Thank you for coming.
Gracias. Thank you very much for having me. Thank you very much to Eduardo for letting me come to present this. So this is a tale of disappointment in part, and so I'll make that clear. So I'm going to discuss some of the studies that we're running. We have a lab. We look at various things, look at multiple sclerosis, but also at stress. And so here it's really focusing on some of the stress studies we've been running recently. So I'll talk about some of the behavioral studies and then some of the larger neuroimaging studies that we've been running and how that leads to Meg's work here with Eduardo. So first of all, we can think of how stress affects us anecdotally. We all know that stress affects our memory. This is what I'm particularly interested in, is how does stress affect your memory? And so if you think back to the times when this has happened, you think back to perhaps when you have to give a talk and you suddenly forget what you were going to say. But you also think about how you can learn very rapidly. So in this case, you have a postman who delivers his mail. He gets attacked by a dog. He's going to hopefully remember this location has this danger there. And so there should be this fast single trial learning of these particular situations. So in this sense, stress is affecting your encoding. You also have stage fright. So here you're supposed to stand up and remember something. Could be when I was playing saxophone as a child and suddenly you can't play what you should know. And so this suggests that stress must be able to affect our memory retrieval. So anecdotally, this all makes sense. It's all intuitive. And so what we're trying to do then is to demonstrate this scientifically in a repeatable way using methods that you can apply time and time again. So the methods that we tend to use in our lab is we use tests of psychosocial stress. So if you are told that you have to give a presentation in front of a group of people, usually a panel, who are sitting there and they're not providing these normal social cues, this threat of peer evaluation triggers an increase in cortisol. It triggers these changes related to stress. And so we do this. This is called the Trier Social Stress Test. You can also use physiological stressors. So a common one is the cold presser, which is where you have to put your hand into ice cold water. And so you see this triggers this cortisol response and so on. So we can use these methods. And from this, what we're trying to do is to see how this affects memory function. And so what I'm going to present to you first is a very cherry-picked version of what we hope the science will show. And this should relate to these changes in encoding and retrieval. So here's a nice study looking at how stress affects encoding. This is by Guillermo Fernandez. And so this was an fMRI study, but we're only considering the behavioral aspects. So in this, participants were shown some film clips, either very violent film clips or some neutral film clips. And afterwards, they were shown some pictures. So a very simple paradigm. And so you have these, put it here, these movie clips. And so they had this film called Irreversible, which is a French film, very difficult to watch film. And so they would watch this, they'd then see these pictures, or they'd have a control condition where they watch just a very neutral film and they see pictures afterwards. What they find from this, if we look here, is you have differences in cortisol levels. So the violent films are triggering an increase in cortisol. You're showing this effect. If you look at pupil dilation, you see changes, greater changes in the stressful film as well. So it seems to be having an effect on the stress system, on the HPA response. And so what you see is 24 hours later, if you look at memory for these pictures, then you have better memory for the pictures after the stressful film than after the neutral film. So stress seems to affect encoding. But I have left this little cherry that's been picked here, because remember, I'm being very selective in the studies. If we think about stress after consolidation, so this is a study that used physiological stress by Cahill, quite an old study. And so what they did here was they showed both neutral pictures and emotional pictures. And then after that, the hand was plunged into cold water, and you look after a delay at memory for these pictures. And what you see is that if you're looking at emotional pictures, there was an advantage for memory of those emotional pictures after this delay. So if you have stress after learning, it influences your consolidation. So James Gough's work has looked at the role of the amygdala in tagging these kind of events and making sure they are better remembered. If we then look at psychosocial stress at retrieval then, and so this was a study by Guez et al. And so here they had 48 healthy subjects, and here they were looking at memory both for items and for paired items as well, of words and pictures. And so these were learnt and then they were retrieved either at the time of stress during this Trier task or during a control task. And so what they found is that if you look at memory when it's retrieved under this kind of stressful condition, you have much worse retention. So this seems to fit this intuitive understanding of stress that I was talking about. So it affects your encoding, it affects your consolidation, it perhaps affects your retrieval as well. So that was the selective version. And so what we want to do now is to think, well, many studies have been running, looking at this in humans. And so what do we actually see if we take all the available evidence? And so this was carried out by Andy Ioannidis' lab in Shields. And so here they were looking at all kinds of different stressors. So physical stressors, psychosocial stressors, people jumping out of airplanes, looking at how that influences their memory. And it was looking at encoding, consolidation, and retrieval. And so what they found is that if you look at all of these different studies, if you look at how it affects your encoding, actually there's a lot of variance, there's a lot of noise in what's coming across. And so overall you get no effective stress on encoding. So this kind of selective picture, this cherry-picked picture is starting to unravel a little bit. When they look at the effect on consolidation, so this is if you're stressed just after learning something, you find that there is a mild effect. It's coming in there. The effects were stronger in men, but there is an effect. If you look at stress on retrieval, so you look at the overall effect down at the bottom here, there seemed to be an effect of stress on trying to retrieve information. You had this negative impact, and this seemed to be stronger when you're using emotional material. So, in some sense, the stage is set. So I had a PhD student. We wanted to look what particular forms of memory are affected by this. So we wanted to look at whether there's differences in recollection and familiarity memory. So the way of thinking of this is sometimes you see somebody on a bus and you think, I know this person. And you don't know where you know that person from, but you recognize the face. And so this is an example of familiarity memory. There could be other times where you see the same person and you think, that's my butcher. So you remember the entire contextual information of where you know that person from. And so this is the difference between familiarity on the first part and recollection. It's something that William James talked about in his Principles of Psychology. It's been looked at by John Aggleton in animal models. And the idea is that where you have recollection, you're remembering all this contextual information. It's hippocampally dependent. And so we wanted to get at, can we actually look at differences in recollection and familiarity behaviorally? And then can we take that to fMRI studies and try and see these kind of associations and activity in these various kind of brain regions? So we ran 4 studies, and we looked using this Trier to try and identify, does stress affect item memory? And so we can separate memory for familiarity and recollection. We then carried out the exact same paradigm. We're trying to look at if stress after encoding affects paired items. So can you remember not just the item, the one it was paired with, but so this would be additional evidence that recollection is affected. We wanted to see how stress affects encoding on emotional memory, because this seemed from the Shields study to be important, and then stress at retrieval on neutral item memory. So here looking at whether stress affects retrieval. So each time we kept the procedure exactly the same, we just manipulated our variable of interest. And so this is a bit small to see, but what we have with this is the participant, when we're looking at post-encoding stress, is that there somewhere? Okay. So yeah, the participant comes in, they have a rest period. We ask them how anxious they're feeling. We take a saliva sample. They then go through a learning task where they learn the stimuli, and then they're introduced to the Trier test. So we said, you're going to run through a talk. You have to present for a job in the department. You have 5 minutes to prepare. You can't take your notes in with you, and so on. They then come in and start that talk. They give another anxiety rating. They have to talk for 5 minutes. And then they do a maths task, which is where you have to count backwards from 1647 in 13s. And if they get it wrong, you say stop, start again. So it's a kind of forced failure task. And then after that, they give another anxiety rating, another saliva sample, and then they rest. And then the next day, they would come back and we look at their memory for these for this material. And so if we look across these studies, we can see that you have an increase in anxiety ratings. So it starts off fairly low levels of anxiety, it increases, and then after the task, it comes down. If we compare that, we also ran, in half of the subjects, we ran a kind of control task. So the same kind of thing, the same activities, but without that kind of stressful element. So they're not being evaluated, and the maths isn't difficult. If we look at their cortisol levels as well, we can see that we get an increase in cortisol in the stress condition compared to the control condition. So it seems to have the desired effects. We're getting these physiological responses that we'd expect to see. If we look at for the last experiment there, this is where we're looking at stress immediately before retrieval. So they would come in on day one, they would carry out this learning task, and then they'd go through the same kind of Trier procedure and then look at their retrieval after all that is done. So this is where the disappointment starts to come in. So everything was set up. And so what we found is that there was no effects of stress on recognition, no effects on familiarity, no effects on recollection. There was a correlation between the amount of cortisol that was expressed and recollection, but there was no overall effects there. If we go to study 2, stress after encoding and cued pair recall, so they had one item.
I have a question about the controls. How were they treated?
They were treated, so everything was done exactly the same.
But they didn't do the presentation?
They had a conversation with the people that were there, but there was no evaluation of it, and there was normal social responses. So there was no kind of peer evaluation. Yeah, so we tried to keep as much as, the same as possible. And so when we did this, again, we had no effect on item recall or cued recall. When we looked at emotional memory, if we were using emotional material, we found no effects of stress and memory for emotional words. And so this is whether we were looking at item performance, whether we were looking at familiarity or recollection, and also no effect of stress on retrieval as well. Here, higher anxiety was related to recollection. So, I'm sorry.
They knew there was a memory study?
No. So it was incidental. So we presented that stimuli, and then it was just presented at the end. And so this is where things were unraveling a little bit. And so we find very limited, weak, non-significant effects. And it's important to say that when the participants were running through this, some of them were visibly distressed, but the memory was still okay. It was still working reasonably. There was some evidence, though, that some of the people who showed greater physiological response were showing more of a change in recollection. So one of the questions is it to do with perhaps the type of stressor, or is it to do with the type of person? Is there individual differences that seems to come in here. And so what we did then is we reran the meta-analysis. So we went back and we only focused on psychosocial stress studies. So from that Shields review where they had all of these different types of stressors, we said, what if you just look at the Trier tasks and similar psychosocial stressors? If you do this and look at encoding, stress after retrieval, and stress at retrieval, do you still see these effects? And so, as with the kind of original Shields study, you don't see an overall effect if you look at encoding. Stress after learning, so if you carry that out and you're looking at how it affects consolidation in human studies and psychosocial stress studies, we didn't find a significant effect if you look after a very short kind of interval, but you do get a bit of an effect if you look after a long interval. And then if you're looking at stress at retrieval, you don't get an effect there. If we reanalyze the studies that were included in Shields, the same thing comes out. You don't see these effects coming out from psychosocial stress. So far, we've said that anecdotally, stress must affect memory. We know it does, but it's surprisingly difficult to detect experimentally. And so this could be, maybe you need to have something that shocks the system, maybe you need to have some pain element that's involved in it, or maybe there are critical individual difference factors, like a past history of stress that comes into this. And so what we wanted to do then was to say, well, if we put the experimental studies to the side a little bit, and then move to very large data sets, can we start seeing this association between between stress, like a background history of stress, and cognitive function, and also can we try and detect brain changes? So I can get this running. So we went to data from the UK Biobank here. Perfect. So in these kind of studies, we're talking about tens of people, 100 people. In a typical fMRI study, you'd be talking about maybe 125 people. So to give an idea of how the scale changes as we move to the biobank, a large study would be about 1,000 people, but here what we're talking about is 500,000 people. So we're massively increasing the sample sizes that we're looking at, and the idea behind the biobank is that if you have this huge sample of participants, some of those are going to develop ALS over 5 years, some of them are going to develop Alzheimer's over 5 years, and so you can track and see what are the early biomarkers there. You can also use it to try and look at hippocampal volumes and linked to aging and also to background history, and so this is where we wanted to use this. So what we did, we took the UK Biobank and we used the childhood trauma questionnaire. And so there are 5 questions from that we can use for this. And so this is looking at whether people have a background history of being physically abused as a child, sexually abused, whether they had somebody who would take them to the doctor if they needed this, and were neglected and so on. You also had questions that would look at recent adulthood stressors as well, so whether they're in relationships where there's domestic violence involved, whether people are out of work when they would normally be employed, and so on. And so from the original set of participants, we first look at those that actually have cognitive data, those that have imaging data, and we can look at people who have low levels of childhood stress and low levels of adulthood stress. So this is pretty much our control group. And then we can look at people who have high levels of childhood stress, high levels of adulthood stress, and also people that had high childhood stress but low adulthood, or high adulthood but low childhood stress. So we can get these kind of different groups and we can start comparing the cognitive performance, we can start looking at brain changes associated with this. From the biobank, there are 2 key kind of cognitive tasks that we could use. First is trail making, and so trail making has a simple version, which is just going from 1 to 2, 3, 4, 5, and so you just need to draw lines between this. And then a second task which involves working memory. And so this is the alphanumeric, so going 1 to A, 2 to B, 3 to C, and so on. And so there you have to like retain exactly where you are in this sequence. There's the digit symbol substitution task, and so this is where you have numbers, you have symbols below, and you just need to put the symbol below each number. So this is a test of both processing speed and also working memory. So looking first at the cognitive effects, what we found is that a combination of high childhood and high adulthood stress predicted poor cognitive outcomes, slightly worse performance, and so you can see that the high childhood and adulthood stress group is doing worse in these tasks than the control group, and that's both on the symbol digit substitution task and on the trail making task that involves this kind of working memory component. We then wanted to look at structural effects as well. So we looked at the volume of limbic structures of the hippocampus, the amygdala, the thalamus. And so this is using the IDPs.
Are you controlling for level of education?
We found that the levels of education were actually similar between the groups, but there was, I think, there was an effect. No, we didn't. I don't think we did. No, we didn't control for levels of education in that one, actually.
It sounds like maybe people who have higher levels of stress would also have less levels of education, maybe because socioeconomic status.
Yeah.
Or something. I don't know.
Yeah, we do. So there is another student who is looking at like what is the relationship with socioeconomic status and using that mediation model. But for this one, we were just looking, whether the presence of kind of stressful background and current stress influences it. So yeah, for this, we were just looking at that in kind of plain models. That's a good question. Yeah. So we're looking at the volume of these structures. We're then also looking at white matter microstructure as well. So we have the diffusion imaging from it, and so we were looking at the DTI, the diffusion tensor imaging metrics. So this is fractional anisotropy, which is if you take your voxels in the brain and you have primarily kind of white matter, does the water in each voxel flow in a kind of directional way, which would be high fractional anisotropy, and so the water is going to be constrained by myelin, by axon membranes, and so on. You have mean diffusivity, which is how much diffusion do you have, and you also have, from a different model called NODI, you have orientational dispersion. And so this is thinking about how many different tracked directions you have within a voxel. And so for this, we were concentrating on the limbic structures, so looking at white matter. So that was within the cingulum of the hippocampus and the posterior thalamic radiation. So we didn't find any group differences in volume, so there was no differences in the hippocampal volume, amygdala volume, thalamic volume between those groups. We did see differences in white matter microstructure, so you see changes in FA between the groups with high levels of childhood and adulthood stress and the effective controls there. And then we're interested, well, is this like a mediating factor? And so we tested mediation models. And so for this, rather than use these kind of groups, we took the entire data set. So we have 64,000 males, 81,000 females. And so we analyzed males and females separately to account for differences in brain volume and so on. And so for males, we found that this kind of orientation dispersion in the posterior thalamic radiation was a mediator of the relationship between adulthood stress and cognition. For females, FA in the same tracks, these posterior thalamic radiation mediated the relationship between childhood stress and cognition. And cognition here is the performance on the alphanumeric trails and symbol digit tasks. So you see that on all of them, and this is correcting for false discovery rate. So we find that both cognition and white matter microstructure were linked to a history of stressful backgrounds. There was evidence that white matter changes may mediate the links between stress and cognitive performance. And there seem to be gender differences in that. And so maybe this is because you see differences in the type of stressors that men and women experience. You tend to see more higher incidence of sexual abuse in women and physical abuse in men. That could come into it, could be cultural factors as well. But we found that the hippocampal volume wasn't related to stress, which is a little bit surprising because there are a number of studies that do show that you get atrophy following childhood stress in the hippocampus. You have this study that shows that childhood abuse is linked to hippocampal atrophy in men, but not in women. And so we took the hippocampus, we got the raw images, and we carried out analysis of subfields, different parts of the hippocampus there. So we used the same group, childhood and adulthood stress, and we split them by sex, look at males and females separately. We used the diffusion tensor imaging and Noddy metrics, but also a different model that I was involved with a lifetime ago called diffusion orientational complexity. And so this just looks at the probability of there being 1, 2, 3, or more than 3 diffusion orientations within voxels. And so the idea is that where you have changes in the composition of voxels, and so that could be because you have glial changes or you have changes in neurons and dendrites and so on, then that would show up on these kind of metrics. And so yeah, so we're looking in this at the head, the body, and the tail of the hippocampus to try and get a better idea of where we see these changes. So as with the earlier study, we don't see any differences in the hippocampus for male and female groups in the volume of the hippocampus. But what we see is that in males in particular, childhood stress was linked to microstructural changes in the head of the hippocampus. And we see this on mean diffusivity, on fractional anisotropy, and in the diffusion orientational complexity. So there seems to be regional differences within the hippocampus relating to childhood stress. So this seems to be gender specific. It was seen in males and not females, and this relates to some of the literature that's come out in the past. We see this in very large samples as well. But it puts forward new questions in some sense. And so what is it? What is it about these particular regions that seems to be affected? Is there a vulnerability of certain neurons or certain cell types to stress? And so one question we're looking at now is, are the regions that do show these changes, do they show high expression of specific neuromodulators? Is there something that seems to explain why we might get these kind of stress changes? And so this is the work that Meg is looking at in humans using things like the biobank, but it's where we can also use translational models and look at these rodent models of stress to see whether there might be particular regions of the amygdala or the hippocampus that are affected. So overall summary, acute effects of stress in humans, particularly psychosocial stress, are actually quite hard to detect. And so we're running studies now where we're taking the same participants, we're putting them through psychosocial stress, and then we're putting them through physiological stress to see, do you actually get differences if you use the same participants rather than comparing, apples and oranges, different studies to try and determine this. There's increasing evidence that background history of stress may be a better predictor of cognitive changes, that individual difference factors come into this. We see changes in white matter and limbic white matter, and there are regional differences in stress effects on grey matter structure, which may relate to gender. And so the question is, why do we get these kind of regional variations now? And so acknowledgement, so this is part of my lab here, and so we have Liz, who's wearing an elf hat at the back, who's carried out quite a bit of this work. We have Meg, who is also wearing an elf hat, and we'll be here for a couple of weeks working. And I'd like to thank you all for listening, so thank you very much.
Thank you.
you for this seminar. I wonder whether the sensitivity of hippocampus is related to connections also, because areas of the hippocampus are more connected to the amygdala,
Yeah.
and then can receive more stimulation from there,
Yeah.
or the connection with the prefrontal cortex.
Yeah.
Apart from what you mentioned,
Yeah.
could be astrocytes, the type of neurons,
Yeah.
microglia,
Yeah.
etc.
So this is what we're particularly interested in, is norepinephrine networks. So do you see, you know, a higher gene expression of norepinephrine gene subunits in this kind of anterior part? I mean, we know that this network does connect to the amygdala, to anterior hippocampus, to prefrontal regions, and so on. And so we're trying to get at that. We haven't looked at, you know, functional connectivity data as part of this, but it's you know, why are these particular regions affected? And so far we haven't seen that it's, you know, specifically norepinephrine areas, but it could be that, you know, there's a combination of neuromodulators within these kind of regions that seem to predict this vulnerability.
Thank you for your talk. Beautiful talk.
Thanks.
Strange, but beautiful.
Strange, yes, yeah.
Yes. These results in general at variance with a whole big body of literature in the rodents where stress interferes with learning and memory.
Yeah.
Can you elaborate that?
Yeah. And so, you know, I think starting off, you know, we took McGough's work, we took, you know, various rodent models, and we're really expecting to see these kind of changes. But I mean, one thing could be about whether there's pain involved. You know, it could be that you need to have slightly different systems that are involved. Like the rodent models tend to involve a physical aspect of stress often. And so this is where we were wanting to compare this in kind of future studies to see, you know, is there a particular type of stressor that you need in order to try and get similar results to the rodent models. I mean, the other thing is, do you need to have a very extreme stressor in humans in order to try and get these kind of responses? We're at a bit of a loss right now, and you know, it should be that if you stress somebody out psychosocially so that they are showing this kind of increase in cortisol, so that they're showing this autonomic response, that you should get these kind of behavioral effects, but they're much weaker than we'd expect to see.
Hi, thank you for your talk. I wanted to ask you something about the UK data set.
Yeah.
I don't know if there is a diversity in the ethnicity. I don't know if they are all, I don't know, European descent.
Yeah. So this is a very good question. So it tends to be relatively homogeneous. In that the people that put themselves, so you know, there's a number of different sites located throughout the UK where they tried to recruit people, and so they have you know, a huge sample, but it tends to be people from slightly higher kind of socioeconomic status that will put themselves forward for this study. You know, if you look at the ethnicity in there, it tends to be white Caucasian that are in there. And so this has been flagged up as one of the problems that we have, not just in this study, but in, you know, whole sets of research that are trying to determine general effects from looking at actually quite specific data sets. And so there is an initiative both by the, you know, some of the researchers that are involved in the UK Biobank, so Carla Miller and people who are trying to get data from sites in Asia, and sites in kind of mainland Europe. And if there are initiatives in Latin America and various other parts, then this is what they're pushing for. I think there's also North Africa. They're trying to build up a data set there. So I think people are very aware that actually, there are problems. There are problems with this data set, but it is very big, and there are different subpopulations kind of within that. But yeah.
Hi, thank you for your talk. This is a very interesting point. I was not going to ask about that, but yeah, very interesting, very good question. So my question actually is because what you showed was about the differences between groups, but what about the variability within the group, like the subjects that had stressful lives when they were kids, and then is there any association with those that showed higher stressors, maybe they showed also greater differences? Yeah.
So when we carried out the mediation models, this is where we see that both the level of stress is linked to the cognitive changes, and also that the white matter changes that we were seeing seem to mediate that. So we did see that relationship, but I think a priori we decided to try and focus on these specific groups that were 2 standard deviations above and both the others. So yeah, we do see that as well.
Just sorry.
Yeah.
Okay. Yeah. Because those are like the subjects with the highest differences, right? So
yeah.
What about the normal? Okay.
Yeah. No. So I mean, the normal group actually were people that scored almost 0 on childhood stress, and so it was actually very flat. And so this is kind of one of the problems in that, is that you just don't have that kind of variation in that. And so it would tend to be that you had a huge number of people who just weren't reporting these kind of childhood stressors, and then people who were reporting actually quite substantial levels, going from small numbers to quite substantial. So the variance, wasn't there for it. But yeah, that's a good question. Yeah.
I was wondering, maybe, what about chronic current stress, like say psychosocial stress, that the type of stress that you have when you're a student or a PhD student.
Yeah.
Every day you have a high level of stress, and that's continuing for a semester.
Yeah.
And that's how my point is, maybe that chronic stress, that levels of stress, is what would show the interference in cognition or
cognitive change.
learning that you were looking well.
Yeah, and in fact, it might even be the other way around. That actually we are carrying out our experiments on students who are used to going through exams, who are used to actually doing quite well in exams because they're getting into university and all the rest of it. And so maybe if we take people, who perhaps aren't as resilient, who have shown more difficulties, then you start seeing these big cognitive effects coming out. I mean, there are many studies showing these inflammatory changes that you get in medical students just before they go for exams. But cognitively, actually, the effects aren't as great as you'd imagine, and maybe it is that actually this is a selected pool. Yeah.
Thanks.
I have a question related to the biobank.
Yeah.
Is there any people that develop any disorder like depression, anxiety, or something like that?
Yeah, it's common.
And
Yeah.
And in the analysis, you control it by.
So for this, we were specifically just looking at this. So we weren't building kind of more sophisticated regression models where we're taking into account anxiety and mood. We did actually look at the prevalence of mental health disorders. So actually, this is counted it's all kind of pooled together. So whether people have anxiety, depression, bipolar disorder, there's a whole kind of different range of DSM categories that are in this. And so it's all kind of pooled. And we did see, I didn't present that here, but we did see associations between childhood stress and the prevalence of mental health disorders kind of later on in life. But we didn't correct for that in these particular kind of analyses. Yeah.
Also, another question. Do you have any report what kind of stress they have the people with high childhood stress and actual?
Yeah. So we have there were 5. What was it? There was 5. Let me pull over here. Try and find where my cursor is. Is it coming in? Okay, I need it to be over on this side. Perfect. So the questions were. Yeah, so the CTQ questions were felt hated by family members, physically abused, felt loved, sexually molested as a child, or had someone to take to a doctor. And so those were the 5 questions that they had as part of the biobank. And then for adulthood, it was belittled by your partner, being in a confiding relationship as an adult, physical partner violence, sexual abuse by partner, unable to pay the rent, the mortgage, and so on. So these are the ones that came out. But we didn't separate them in this particular study. We didn't see, do some of those have a greater effect than others, which is one important point. Which is important, and also could play in, as I was saying, it could play into these gender differences as well. Some of those are overexpressed in some than others. Yeah.
I know, but I'm very aware that what you just showed us is the first approximation, something that is more elusive. I'm sort of skeptical of the strategy of you have an elusive behavioral phenomenon, and the next step is to look around in a big, flashy biobank if something relates to what you find in the missing stuff, in the behavioral stuff. So I'm a little bit skeptical of that strategy.
Yeah.
What I was thinking about is if they have ever tried of looking for someone or some group of people that are very prone to stress,
yeah,
and making them learn to act differently. And
yeah,
to see if maybe perhaps there's some relationship in some structures, like the campus, the amygdala. I don't know
yeah,
if that's been done right.
Yeah. So we're doing this where we're actually screening people first to try and identify these high and low groups, and then carrying out the experimental studies. And so Rebecca Elliott in Manchester has been running these studies to try and look at these differences that you might get, but not with the Trier test yet. And so yeah, it's a program of research where we had one particular student who is carrying out these experimental studies, and then trying to scale up a bit to see whether you get these effects. And then once you know that certain things, that relationship exists, you then go back and then try and actually locate those people and then run the experimental studies to see whether these effects come out. So yeah, they're quite different studies in some sense, but it's trying to get at where do we actually start getting this. Yeah. Fair.
I would like to ask about the treatment. I mean, if in the adult population that you have been studying in the UK data, you saw any difference, or you evaluate about the treatment they have before the resonance, because we are going to have some difference if they have, for example, any kind of mental illness with treatment in those gray matter or white matter.
Yeah. So this this was just using the kind of existing data set, and so it didn't account for a lifetime treatments, both pharmacological treatments and also behavioral treatments that come in there. We only have data, like recent data as well, so you don't have a full kind of medical history for each kind of individual that goes in there. So you'd be able to get some kind of information on whether people are currently on medication, but not like anything going back a little bit. And so
sorry, but there is possibility to see in that data if they have for example, in childhood, this kind of medication for any kind of illness?
You would only be able to get their current medication, so their current treatment that they have from this. So there are going to be a lot of gaps on this, and you're kind of using the scale of data to try and get away from some of these problems. But yeah, I mean, you know, this is the problem with the biobank, is that it doesn't contain the tasks that you want to run. It contains very basic tasks, and it doesn't have the detailed information on participants that you perhaps want for your studies. So you have to treat it as a separate way of complementing your research studies, but it's not a substitute for it. You know, if you have a particular targeted question, you need to carry that out.
Can I just build on that really quickly? Something that Biobank are going to start doing from next year, I think it is they're incorporating like local GP records. So they're trying to get in contact with the NHS and get like previous medical history records. So the idea is that they'll be able to see all of the things that they've been treated for, all the things they've been diagnosed with from pretty much birth or a very young age, to then incorporate that onto the biobank. But for 500,000 people, that takes a very long time. So they're doing the first bit of that now, so that information should be available on biobank next year or so, but at least a subset of people. Yeah, it should be really good, especially when we're talking about identifying like biomarkers and things like that for disease, because you can see, you know, previous medication they've been on, previous treatments they've gone through, etc. So it should open some new doors with that as well. It should be really good.
The other thing that comes in as well is that they're doing this kind of as a kind of longitudinal study as well. So people are going to be rescanned at various time points, so you can see whether those baseline factors predict actually how people go on to evolve and develop conditions and so on.
There are no more questions, then thank you. Thanks again.
All right, thank you.